Hi everyone, welcome to our new video. This time we will start discussing about the morphological identification of chica mites. In this first series of identification, we will start with chicker's mouthpart, or also known as gnathosoma. Chickers are often found in clusters. This figure depicts clusters of chica mites inside a rodent's earlobe. Due to the very small size of chica mites, which are generally less than 1 millimeter, the morphological identification can be quite difficult. There are several organs that are important for chica mite identification up to the species level. These organs include the scutum or the hard plate on the dorsal part of the chicker, the dorsal setae, the legs including the claws and specialized setae, and the mouthpart or gnathosoma, which consists of chelicerae, galliali, and palpi. In this video, we will focus on the mouthpart. In the upcoming slides, we will discuss each component of the mouthparts one by one. This figure shows the visualization of gnathosoma from the dorsal and the ventral side. First, let's talk about the chelicerae blade. In conjunction with other morphological characters, these can be used to help identify chickers up to the species level. These chelicerae blades have distinctive characteristics in different species. In Radfordiana nudosita, as shown in this slide, the chelicerae blade is long and slender with 1 ventral and 8 to 11 dorsal teeth. In Schoengastia roselli, the chelicerae blade has 1 ventral and 13 to 15 recurved dorsal teeth. In Odontosaurus unisetosa, the chelicerae blade has 5 to 6 dorsal and 5 to 7 ventral teeth. In Xenodontosaurus serratus, the chelicerae blade has 12 dorsal, 13 ventral, and 2 dorsolateral teeth. In Honeymannia chaparensis, the chelicerae blade is on the outer side with a row of about 8 teeth. In Bamerella latiscuta, the chelicerae blade has an expanded hood-like cap bearing marginal denticles. In Ascoschengastia gopti, the chelicerae blade has a tricuspid cap. In Babiangia danieli, the chelicerae blade has butting tooth and a lateral hook. In Ernoschoengastia cana, the chelicerae blade has multidentated apex and showing no definite tricuspid cap. In Walchiella odomensi, the chelicerae blade has 3 widely set recurved teeth and a tricuspid cap. In Eutrombicula hirsti, the chelicerae blade is long with a tricuspid cap. In Novotrombicula oviensis, the chelicerae blades are long and strong. Beside the chelicerae blades, the gallial setae is also an important morphological character for chica mite identification. The nude or branched gallial setae can assist in the genus identification. In some species-level identification, the number of branches on the gallial setae is used to classify the species. The description of the palpal setae on the palpi can be presented using the following order. Starting with the palpi on the dorsal side, the first is the femoral setae. The second is the genual setae. The third is the tibial setae, with the order of the dorsal, lateral, and ventral side. Looking from the ventral side of the tarsus part, we describe the number of the nude or branched palpal setae. Finally, we describe the presence or absence of the subterminala. This example shows the description of the palpal setae. First, we have a branched femoral seta. Second, we have a nude genual seta. Third, we have a nude dorsotibial seta, a nude dorsolateral seta, and a branched ventral tibial seta. Fourth, we have a 5-branched palpal tarsus setae. And lastly, fifth, we have a subterminala. Now that we have described this, let's go on to the palpal claw. After describing the cheliceral blade, the Gallial setae, and the palpal setae, we will now move on to the last part of the chicker's mouthpart, which is the palpal claw. Here, we need to see the number of prongs. This character can assist in morphological identification for up to the species level. In Rundiphodia nudosita, for example, the palpal claw is simple. In Schoengastia roselli, the palpal claw has 2 prongs, with the outer prongs smaller than the inner ones. In Cynodontosaurus serratus, the palpal claw has 3 prongs. In Odontosaurus unisetosa, the palpal claw has 4 prongs. In Odontosaurus riani, the palpal claw has 7 prongs. In Eutrombicula hirsti, the palpal claw has 2 prongs, with the outer prong bigger than the inner one. And in Babiangia danieli, the palpal claw is bifid. In summary, the organs of the mouthpart or gnathosoma that can assist in chicker mites' morphological identification includes the cheliceral blade, the Gallial setae, the palpal setae, and the palpal claw. These organs, in combination with other morphological characters, can provide an accurate identification of the chicker mite species. Several identification keys were used as guidance in constructing this video. Finally, we would like to thank our institutions, collaborators, and everyone who supported this project. Thank you for your attention, and we'll hope to see you again in the next video on the theory of chicker mite morphological identification.